So I'm Mark Chaisson. I'm assistant professor at USC. So this will be the second talk I'm giving through here, but I'm not really going to be around for too much this week. So if you want to chat about any of this, just send me an email and I'll head by. So I'm going to talk today about a project that I worked on largely up in Washington as a postdoc, and I'm going to start to kind of continue a little bit of it down here in my lab. And it's about this kind of what we see as the sort of next step in the frontier of doing de novo assembly of human genomes. So Mike Schatz gave a great review on de novo assembly last week. So the whole goal is to take a set of reads that have been sampled from this shredded genome and try and reconstruct the genome. And there's a few different formulations where you can have either overlap, De Bruijn, or string graph representations of the assemblies. But all of the different representations have the same problem or same dilemma, where if reads aren't able to traverse an entire repeat within the genome, you're left with a fragmented assembly, and you're not able to reconstruct the entire genome. What's shown here is this typical toy example where there's two repeats and a set of fragments from the repeats, and no fragment spans it. And so the assemblies that are left over are the unique regions of the genome, and then what's kind of called a collapse of the repetitive sequence in between. And just how that collapse looks is a little bit different depending on the graphical representations, but the similarities are that there's no read that traverses all of it. So we're able to resolve much larger duplications by applying assembly with longer reads. This is a slide that I showed, or a figure from a slide that I showed last week, and it's the statistics for long read sequencers for two recent runs that were done, one on Oxford Nanopore, one on Pacific Biosciences. It's just showing that the reads are of order of length of 30 to up to 100,000 base pairs. And what this means is that we can start to try and resolve duplication copies within genomes that are of that length, so 30 to 100,000 bases. There's been a lot of work that's been going on into doing de novo assembly with long read sequencers. And so it really started in about 2015 with the MHAP aligner by Konstantin Berlin and Adam Felipe, where they developed a very fast but sensitive overlapper that was able to perform the pairwise alignments in these very large-scale single-molecule sequencing data sets, and it essentially solved the problem of how do you go about finding all pairwise alignments within a very noisy set of fairly long sequencing reads. And then this has kind of been refined with different methods that have either applied different approaches to finding pairwise alignments or different embeddings of those alignments into different graph structures. And this most recent work that was done is I believe, submitted. At least it was shown in Recom last year, and it was an embedding of pairwise alignments in something called an De Bruijn graph, which you could kind of think of similar as the string graphs from some of the original Falcon papers. Falcon being another long read alignment method. And if you look at the contiguity of assemblies that have been performed with short reads and long reads with kind of improving methods across over time, what you can see is there's been an exponential increase in contiguity of human genome assemblies over time through both a combination of improved algorithms and improved data structures. But they all have something in common where there's a bit of a drawback. And if we map the contigs that are generated from a recent assembly of a Puerto Rican individual, this assembly had an N50 of around 16 megabases, so this corresponds to a highly contiguous assembly. What's shown in black are long contigs over 100 kilobases. In blue are the short contigs, so more fragmented contigs under 100 kilobases. And in red is shown segmental duplications that are over 50 kilobases. And so just visually you can clearly see that there's this pattern where when there are segmental duplications that corresponds to the fragmented contigs in de novo assemblies, even using long single molecule sequencing reads. So again, this is another slide that I showed last week, but to kind of go over it again, you can take de novo assemblies and try and measure the contiguity with respect to resolving segmental duplications. And so mapping these contigs back to the genome, we can look for annotated segmental duplications. So it's shown in green as an annotated segmental duplication. And if a read spans, if an assembly spans plus and minus 50 kilobases over a segmental duplication, we consider that to be resolved. And if it ends kind of in the middle of it, we consider that to be unresolved. And this is important because if it spans the entire duplication, that means we've resolved what the actual structure is and not just sequence content. And that's important that I'll show in a couple slides later. But what you can see is in red are shown the unresolved segmental duplications in an assembly of a hydatidiform mole. So this is a haploid cell line. So it's essentially a much easier case of de novo assembly where you don't have to worry about allelic variation. And still with this haploid cell line, you have an assembly where the duplications that are over 50 kilobases and over about 98% identity are left unresolved. So the question is, how do we go about improving assemblies given this limitation that we have? So unfortunately, one of our recent reviewers said we should just wait and let technology improve and let that go about assembling the contigs. And that's actually, to be honest, it's something to always keep in mind when you're developing ideas for research, is you want to try and target ideas that are going to be relevant a year and a half to 2 years from now. And technology is moving so quickly that it's tough to hear, but sometimes it is a good criticism that maybe the problem that you're working on now might not be so relevant a year or 2 years from now, especially in these technology-driven fields. So I think this problem will be relevant for a little bit of time, and it's something worth working on. And so it's something that we've applied a couple of approaches to. One quick little thing about the biology of segmental duplications. So a segmental duplication is a sequence that's functionally defined as at least a kilobase in length and 90 to 100% identity to another sequence elsewhere in the genome. And it's not composed of simple tandem repeats or transposable elements. So some interesting biology about these is they're actually gene-rich relative to other non-segmental duplication regions of the genome. And so because of this, they are actually involved in many genetic disorders. So different loci that are rich in duplication can be responsible for Williams-Barian, Prader-Willi, Coulian-de Vries, a number of syndromes that happen due to rearrangements that are mediated by segmental duplications. In particular, the specific genetic architecture of segmental duplications can predispose to or against disease. So there's a locus that I like to bring up as a good example. It's the 17q2131 locus, where there is an inversion that's floating around in the population. It's about 900 kilobases. It was shown in 2005 that individuals with this inversion have an increased fecundity, so about a 3% increase in birth rate with individuals with this inversion than without. However, there's also an issue where when you have this inversion, you become predisposed to a microdeletion syndrome. So the Coulian-de Vries syndrome originates from the deletion of the cancel 1 gene in this particular locus. So there's this really interesting evolutionary pressure in this region where there's both a benefit and a drawback to having this inversion. But the key is that you need to know what the architecture is of an individual to know whether or not any of these kind of interesting biological elements apply. So we're doing well with assemblies, but we need to develop new methods that are able to go and essentially resolve this last frontier of genetic architecture. The nice thing about these duplications, I said that they're 90 to 100% identical. They're very rarely exactly 100% identical. So you can think of these duplications as occurring on an evolutionary timescale, although they do happen in essentially de novo or newborns in the population. But if we want to resolve an architecture that's been around for a certain amount of generations, we can think of the duplication as occurring, and then over a certain amount of time, each different copy of the duplication acquires variants. These variants are referred to as paralog-specific variants because they can, each variant is specific to the paralog, obviously. And what's nice about this is they can be used to distinguish the different paralogs during assembly. So they're not so frequent that when detecting overlaps between these long, noisy sequences, you can distinguish the overlaps from different paralogs. But what we're going to look at is, given an assembly, maybe we can go back and try and improve it with methods specifically tailored to resolving duplications. So I showed that earlier cartoon of assemblies. So what's shown here is if we had this as our genome, where we have this red duplication exists 3 times in the genome that's been flanked by unique sequences, we can kind of consider something a generic assembly graph. This isn't really any sort of De Bruijn or string graph of any sorts, but each kind of rectangle corresponds to a sequence, and each edge connects any 2 sequences, and the genome can be spelled out by a path through this graph where you emit the sequence corresponding to any vertex as you pass through it. And so we want to try and resolve the content of this genome. Well, one of the things that we could potentially do is we could look for sequencing reads that traverse a little bit from the contigs flanking this collapsed duplication into the duplication, and maybe start to peel apart the duplication from that. And we tried this for a while, and unfortunately with the assembly graphs that we had, didn't have a good enough representation of the duplication structure in the genome, and we couldn't really figure out a way to in a general manner, kind of work our way into duplications. And so instead we took a different approach, where what we'd like to do is take this collapsed sequence duplication and then try and peel apart the different paralog copies that are inside of the duplication. So the input is an assembly, the sequence of a region that we know to be collapsed, and then a set of reads that we've mapped back to this duplication. So during the assembly process, we can assume that we're either keeping track of this information from the pairwise overlaps, or we can just map reads back to the assembly. And the output that we'd like to have is the set of paralog-specific variants corresponding to the duplications that have been collapsed to this, or equivalently, the consensus sequences of the different paralogs that are collapsed into this one merged copy above. So what does this look like in terms of input to a program that we're writing? Well, we consider the genome as a set of sequences, or a set of nucleotides, and we're only going to care about, the genome in this case just corresponds to the collapsed duplication. And what we're really only interested in are the columns that have variable nucleotides in this, so we can ignore some of these invariant columns. But the reads are essentially a set of nucleotides that are sampled from this genome matrix. So again, the genome is, this is kind of the oracle genome, where it's a set of collapsed duplications. And in the error-free case, we have a set of reads that are essentially just read off as subsets of each row in this genome matrix. What we'd like to have is a partitioning of these rows in the read matrix such that all of the variable nucleotides in any particular column within any partition are in agreement with each other. So if there's no sequencing error, that's a very straightforward task. You can do a multiple sequence alignment, you can scan along the reads, and then you can emit the consensus sequences of the different paralog copies. But unfortunately, we're looking at either PacBio or Oxford Nanopore reads. In the first set of experiments that we did, we've only looked at PacBio reads. And it has a very high error rate, so we can't just do the simple approach where we can do a multiple sequence alignment and use consensus. So when trying to separate paralogs with real sequencing reads, if there's only 2 paralogs, this is actually a well-studied problem. This is just simply the diploid phasing problem. And actually, Vikas Bansal gave a really good talk about this last week. If there's more than two paralogs, then this just corresponds to the polyploid phasing problem. So we'd essentially like to try and separate out the phase of separate paralogs. And so we took two different algorithmic approaches to trying to solve this problem. One referred to as matrix completion with noise, and another that we refer to as correlation clustering. So we'll talk about the first problem. The setup for a matrix completion problem is referred to as, is commonly known as the Netflix problem. So what's going on here, this is kind of an oracle where we know that there exists some true matrix. This true matrix represents the set of all known movies and the set of all users, and it's the preference, the ranking of the user to the movies. And so this is what we'd like to, is some oracle knows, but in reality, no one user has seen all movies, and it's not, and no user is extremely consistent with their ranking of movies. But even with that, what we'd like to do is take the small set of movies that someone's watched, and then try and use that smaller set to try and predict a set of recommendations for those users for the movies that they haven't watched. So the key sort of constraint in this that lets this recommendation happen at all is that we assume that this evaluation matrix is of low rank. And so what's shown here is kind of the equivalent of this, where we can say, okay, there's a lot of different users that are using Netflix, but in reality, there's just two types, one that likes foreign movies and one that likes action movies. And and what you have is each individual user is essentially sampling from this one low rank matrix. So the goal is to fill in the missing values of X such that the rank of X is minimized. And in the Netflix problem, what you can say is the goal is to fill in the values of X such that the rank of X is minimized while preserving the exact preferences of any original individual user. That's not exactly the exact case that we have here, because there's a bit of noise in every selection. So to go into more detail about the approach that we have for polyploid phasing, you have an input matrix G. So this is sort of the oracle of our genome. And what we can do is we can condense this into a matrix that where we ignore all of the invariant positions, and then we can relabel anything as +1 if it's essentially the consensus or the reference, and -1 if it's different. And so this is the matrix of the paralog-specific variants that we'd like to end up recovering. And what we have is a set of fragments. These are the reads that we're getting, and essentially it's just subsampling from this original genome matrix B. And so for every read we have a subsample. The full matrix M, this would be the equivalent of the movies that we'd like to try and kind of reconstruct for recommending, has complete values for every row, but the actual read matrix is something you don't assume that every read will traverse across the entire genome, so we have essentially unknown values in the read matrix. So this is, again, corresponding to the problem of estimating the true read matrix M from a noisy read matrix. So the setup for this is we can assume that this true matrix can be factorized as a M by S matrix where each row is an elementary vector. So it's essentially deciding which copy of a paralog we're selecting, and a set of elements B, which correspond to the copies of the elements of the to the paralog-specific variants from the genome. Oh, let's do that there. Okay, so in order to estimate our true read matrix M, we'd like to optimize this function where what we have is the, we'd like to minimize the difference between the known, the estimated paralog-specific variance, and essentially these indexes into the selecting for which paralog any given read is coming from. And this p omega is a function that essentially masks off all of the unknown values in the read matrix. So this can be found using this. So this is an NP-hard problem in order to do this optimization. So we can split this optimization into two separate steps that are, it's a heuristic that involves two convex optimizations which can be solved in polynomial time, where essentially what we first do is we fix this selection matrix where we're selecting which paralog any given read comes from, and then we estimate what the read matrix is, and then given the read matrix we essentially re-update what the selection matrix is. So this was first solved through structure-constrained gradients descent in 2016, where A is this elementary vector, and B, instead of being a set of integer values for the read matrix, it was not constrained, and so B was this real matrix. So the approach that we have is slightly different. It essentially tries to go from this real valued matrix on the set of reads to forcing it to be integer valued. So the first step that we can do is, given some estimate of the selection matrix and the read matrix at any point in time, we can estimate a real valued read matrix, and then take that real valued read matrix and essentially force that into discrete values for the selection and the reads themselves. Okay, so this quick outline of that is to, there's this first initialization step where you can take the essentially multiple sequence alignment of the reads and use SVD to estimate the first potential set of PSV values, and then go back and iteratively calculate the real valued, the discrete valued selection and read values until there's some form of convergence. So what does this look like? We have this original read matrix X, we have an unknown read matrix A, and then we can use SVD to estimate the first iteration of the potential genome values in B. And given these potential genome values in A, we come up with essentially a maximum likelihood estimate of where any given read was chosen from, and then these values are rounded up to integer values that are then used to re-update the genome matrix. So this does have a bit of a problem where there tends to be certain paralogs that have a very high divergence from the other sets of paralogs. And so we found that it tended to be a bit more accurate if we try and resolve the more divergent paralogs first, and then sort of iteratively remove divergent paralogs until there were essentially no paralogs left. And so there's a slight change in the method where we start with the same initialization, run the optimization, and come up with an estimate for what the genome, what the set of paralogs are in the genome. Remove the best haplotype. And then reiterate with an estimated ploidy that's one size smaller. Yeah.
So on your previous slide, like you know, your you're kind of assuming that all of the reads line up, right? That they're aligned, and like, you know, isn't some of the reads only capture part of the paragraph, right?
Right. So you're not assuming. So this is the read matrix, and you're not assuming that any read traverses the entire genome.
I see.
Yeah.
Okay.
Okay. So the approach using matrix completion needs to essentially estimate the most divergent paralog first, and then keep iterating until there are no paralogs left to separate out. There is a bit of a problem with this formulation, and it's essentially ignoring the fact that we have paralogs that are unique to any specific variant. And so what's shown here is a matrix that's of rank 4 but has no variants unique to a paralog. And this is something that the matrix completion problem is capable of separating out. Whereas here, there is a different matrix that's of rank 4, and each paralog has a unique variant. And this is more akin to the structure of the problem that we have. So there's a different approach that we have that's called correlation clustering, and we use that to take advantage of the fact that we have these paralog-specific variants. So the setup to this problem is to estimate the set of paralog-specific variant sites within the genome. Then we build what's called a paralog-specific variant graph, where each node corresponds to a PSV in the genome, and any edge connects two different PSVs if some sufficient number of reads overlap those PSVs. So because there's noise, what we have are some false positive edges, and maybe we have some missing edges, so it's an incomplete graph relative to kind of an ideal picture of what the different paralogs would support. So in order to try and distinguish these sets of edges that are potentially noisy edges from true edges, what we add are a set of these negative edges, where if we have two paralog-specific variants that are nearby each other in terms of genomic location, we add a negative edge between those two variants. And then what we'd like to do is come up with some sort of clustering that's able to cluster together PSVs from the same paralog, but not include PSVs from different paralogs. And so this is pretty easily stated in the correlation clustering optimization, where what we'd like to do is find a set of clusters. In this particular case, we don't have to have a specific number of clusters that we're looking for, but the clusters optimize the function where we're trying to minimize the number of negative edges that are within any given cluster, plus the number of positive edges that are cut between any two clusters. So this is APX hard. We have to come up with some sort of heuristics to solve this. We used a heuristic that was similar to something that was proposed in 2005, where we essentially pick a high-degree vertex and keep adding its neighbors to its cluster until that cluster grows to a size that, until the score of that cluster stops increasing. And then we actually do this multiple different times to sample different potential clusterings, and once those tend to converge, then we emit those converged clusters as the final result. So to test this, what we did was we took a few different evolutionary processes where we first modeled 14 what we called structured trees, where you had either kind of a symmetric process where the paralog copies are being duplicated, either as this sort of grandparent and children process, or these kind of tree type structures. And essentially, every branch that happens, some unit of a certain percentage of additional variants are added. And that was done for these structured trees. And then we also looked at 50 random trees. And so each random tree had 10 child nodes, but with different randomly chosen topologies. So to evaluate this, we looked at the minimum error correction score between the only existing method that, that worked on this data set, something called structure constrained gradient descent, the matrix completion approach that we had, correlation clustering, and then matrix completion that's seeded with correlation clustering instead of SVD. And the simulations were done on different units of evolutionary time per new generation, between essentially 1% down to 0.5%. And it's a little hard to distinguish the different points that are here, but swaying off the diagonal shows that the minimum error correction is lower on that method versus the other, so you want to be closer to your method. What's actually shown here is that the swaying off the diagonal towards your method is an increased minimum error correction relative to the other method. And so in this particular case, even though the matrix completion problem was able to handle this more, or it was able to handle this more generalized case, where it's not focusing on paralog specific variants, and it actually had, in general, lower minimum error correction scores for across most of the simulations that we did than correlation clustering. However, when we go and try and find out how many different paralog copies are correctly resolved, we found out that the correlation clustering approach, and again, it's a little harder to see here, but what's shown here is for every simulated data set, how many copies on the x-axis were simulated and how many copies on the y-axis were reconstructed. And by and large, the correlation clustering approach was doing a better job at reconstructing the actual paralog specific variants than the matrix completion approach. So this project was essentially just done kind of on simulated data. A student of Evan Eichler has taken this and applied it to real data and trying to resolve collapse assemblies collapse duplications in human assemblies. So he came up with this pipeline that takes a human de novo assembly, maps sequences back to the assembly, and then scans across that and looks for read depth that's kind of spiking up so that it indicates that there's been a collapse duplication in any particular region. Then we just ignore all of the regions that have kind of expected read depth and then focus only on the collapse duplications. Run correlation clustering to sort of develop, see if there's a partitioning that can separate the paralog specific variants. Separate those out and essentially reassemble the clustered reads and then emit those reads as contigs. And the input assemblies that we looked at, we considered 3 different assemblies. The N50s of these assemblies were relatively large compared to a lot of current approaches or a lot of past runs. So between about 27 and 29 megabase N50s. Pretty high coverage, around 60-fold coverage, and read lengths that were fairly long, up to 17 to 20 kilobases. And the amount of unresolved segmental duplications, if we use that metric of plus and minus 50 kilobases on either side of any segmental duplication, was about 125 megabases per assembly. So this is something that we kind of see as fairly constant between any different method that we use and any different data set that we apply, is this kind of same set of duplications that are not resolved. And so after applying correlation clustering, what we saw is that we're able to resolve between about 18 and 40 megabases of additional contigs, an additional sequence that wasn't present in the original assemblies. What's shown here is this diverged amount is the number of bases that we found to be essentially different from the reference and not likely to be, essentially it's new sequence copies that aren't part of the existing reference sequence. And the way that we know that these are actually correctly assembled sequences and not just kind of, if a sequence doesn't exist in the reference, it's either some horrible misassembly that doesn't belong there, or it's a correct assembly that's not present in the genome. And so we compared, I forget the exact number, but on the order of a few hundred of these resolved duplications to back, so bacterial artificial chromosomes that have been sequenced from the CHM1 genome, and showed that sequences that had this really low mapping identity back to the human reference have a nearly 100% identity mapping to these back clones that have been sequenced and kind of hand-finished, and they're sequences that are known to not exist in the standard HG38 assembly. So in summary, SMS, so single molecule sequence assemblies, are limited by segmental duplications in what's kind of modern human assemblies. Segmental duplications do have these infrequent paralog-specific invariants that we can use as small keys to try and distinguish the different copies of a variation. Matrix completion and correlation clustering are fairly effective at partitioning these PSVs, and correlation clustering on real data resolves 18 to 39 megabases of duplicated sequences. And so thank you guys for this, for the chance to give the second talk. And again, this is a really good collaboration with Sriram Kannan, Sudipti Mukherjee, and now Mitchell Volger all up at UW.
I have a question. Maybe it's something that I missed. In the matrix completion kind of strategy, you discretize in every iteration.
Yes.
Does it make sense to maybe continue in the undiscrete
So,
and discretize again?
yeah, so that's what this other structure-constrained gradient descent approach does, is it leaves everything in real until the last step, in which case a rounding is applied. And so then it had slightly worse MEC scores, and then it's, again, it wasn't written for this approach,
Yeah.
but it had really bad results in the actual reconstruction of these variants.
So what's the density of the PSVs? Like, I mean, do you see like one every KB or what?
So there's a pretty huge gradient. So one every KB is about the minimum.
I see.
Yeah.
So if there's like 3 or 4, I mean, could you say, because they exist, could you potentially just use them? Like, I mean, what I'm meaning is that your method captures them plus weaker signals as well.
Mm-hmm.
I'm just saying is that could you just use them
No. So the correlation clustering does just use the PSVs.
without the matrix completion?
Yeah.
I see.
So the matrix completion handles these variants where maybe there's two duplications that have one variant and no other ones of, say, some multi-copy duplication.
So you basically use the PSVs to get you pretty far, and then you use the matrix completion for the rest. Is that kind of?
No, they're just two totally separate approaches.
I see.
So there definitely needs to be some development that happens that pushes it a little bit further.
So you're going to write a new paper about combining the two?
Probably. Yeah. I actually hired I hired someone from the lab who wrote the structure-constrained gradient descent as a postdoc, so she's there to work on that.
Mark, so maybe two separate questions. First, maybe more on the biology of these duplication, how they're created, because I'm interested, and then on more technical. So you had in the matrix completion, you showed it's hard, but then you have this heuristic that you set, you put constant subset of variables and optimize the others. And do that. So this is more a statistical approach, correct? More an EM. So
Yeah.
how these two, so combinatorially it is hard, but then you do some statistical approach. How you combine them, and again the biology behind these duplications.
Yeah.
Okay.
So the biology behind the duplications is that there's a bunch of processes that I'm not super familiar with, even though it's kind of a required thing and as an Eichler Lab postdoc for knowing the biochemistry of how these duplications form. But what tends to happen is there can be something called duplication cores, and these are short, somewhat palindromic sequences that at one point in time emerge as kind of this seed, and this seed primes the existence of other duplications. So once you have one copy of one pair of duplications, then through non-allelic homologous recombination, you tend to grow that into more and more copies of a duplication. So that's why you tend to have these mosaic structures where there's clusters of duplications, and that's because once one happens, it primes the existence of others. So for the matrix completion, so it's a hard problem, but it's very dependent on the initialization of the problem and the fact that most of these are actually fairly easily separable. So there's this picture that was shown, let's see. Okay, so this is actually close to a fairly typical PSV graph, where each different color is kind of the ground truth of the PSV. And so even with fairly simple kind of cutoffs and heuristics, there's not too many edges of these kind of false positive edges that exist between any two clusters. And what this corresponds to in the matrix completion sense formulation is that any two different rows that shouldn't be clustered together have a fairly different difference between the two of them. So it's the problem, the next step in this problem is to start to push into that noise where there's very highly identical duplications and there isn't a lot of differences between any two sequences. But for the time being, the first, say, third to half of the unresolved duplications have a large number of differences between them. Ryan, or?
Yeah, to follow up to this question, also on Cedric's work on this, would you say that if assemblers were more careful in how they overlap reads, they would possibly symmetrical duplications, or there are some other effects?
Probably. So there was some work by Bernard Carvalho who used Illumina sequences to try and find all of the regions of reads that are probably not erroneous, and then only aligned using those more accurate parts of reads. So that worked really well, but then it ended up throwing out so much data that that the assemblies didn't look so good within the duplicated regions, or kind of in general. It's probably likely to be able to go back and do some post-processing of the alignments themselves. So in particular, if you don't look at the edit distance, you look at the edit distance only with respect to SNVs between two sequences. That can probably help at the overlap phase. The problem is that it's hard to have that be perfect. And so anytime you have kind of an imperfection where one read does get mismapped to another read from a different paralog, then that can start to kind of merge back together the assembly graph.
Yeah, so kind of related to previous question. So you mentioned like the error rate. So how important the error rate is? Like, for example, if you suddenly got the long reads which are perfect with no error, would you expect the results?
Then we can go home. Yeah, we don't need to do this.
Because like in that case, do you think it's like combining, like taking the PacBio data or nanopore data and then enhancing that with the Illumina data? Or like trying like there are computational error correction methods which can potentially to draw the error rates of all those ones.
Yeah, so the problem is it's a hard mapping problem to go from an Illumina read to the correct to mapping reads from different paralogs to each other correctly is hard, even when you have something that's as accurate as an Illumina read. So it's possible that it could be done, but it's something that